funny. I sent them a picture of my car yesterday. And then they sent me a picture of like green grass.
Coffee. Tea? Okay. Both, Brandon? Okay. What we're going to do is I'll make a couple of pots every Friday. So it's a BYOM, bring your own mug. But I'll make coffee on Fridays so that we can stay awake for the last hour of class. Okay. All right, so BYOM this Friday. So what I wanted to do today is let's just start by recapping a couple of things that we talked about on Monday, and that is just give me 3 features of any general virus. Okay? So take a minute, jot down a quick list for yourself, 3 features of any general virus. All right, 3 features. Okay. Needs a host cell. Filterable. In other words, this tells us about size, right? Has some sort of genetic material. And so that genetic material, we said there's 7 different types. Well, I guess there's really 2 types. There's DNA and there's RNA. But then when we break it down into, well, is that RNA double-stranded, or is it single-stranded plus, single-stranded minus, so on and so forth. Friday's conversation, we will spend more time talking about that, and then we'll actually revisit it a second time next week and even the week after. This is kind of a main theme that we're going to spend the first few weeks on. Okay, so those are 3 general features. Are we missing anything else from this? Yeah, and we'll come back to that one, and that's kind of the conversation for today is this capsid. And maybe the first question to ask here is, what's the function of a capsid? So if we think about it, Brendan.
Protect the genetic material.
Yeah, that's the main component, right? And so, again, the structure that we're going to talk about, what we envision as a virus, we will call the virion or the viral particle. And the main purpose of that capsid is to protect the genomic material, because outside of the cell, there are a number of nucleases, environmental and physical stressors like UV, heat, that can lead to denaturation and damage of DNA. And so the capsid there protects it from some of these environmental and physical effects. The other thing of the capsid is that it's required for entry into the host cell as well. All capsids are going to have some sort of spike or receptor protein that allows for the virus to enter into the host cell. And so that's where we're going to kind of focus today, is on describing the structure of the capsid and then talking about some of the basic components of it. If you're following along with our list of objectives, the goal today is really to kind of get through the first 2 here. We'll get through the second 2 on Friday. All right. Capsids. A couple of different flavors. In terms of shape, icosahedral, helical, or complex. And then the second feature that we often look at is, are these viral particles enveloped, or are they what we would call naked or non-enveloped? What I'd like you to do, either envision or draw for yourself the following. Draw for yourself a enveloped icosahedral capsid, or virion. These do not need to be spectacular drawings. Okay, you do not need to draw a 3D rendition of this. But roughly, what would this look like? And once you've got that vision in your head, which of these best fits that? So here are 6 different viral structures, or virion morphologies, from different from different viruses. Which of these electron micrographs best fits with the image that you just drew? Annie? Which one?
The one on the bottom.
Bottom in the middle, right here. Okay, so what about this one fits with what you've drawn?
It's kind of spherical.
Yeah, and typically what we'll see with icosahedrals, if we look at them in 2D, they either look spherical, or if you could envision this one, it has kind of a hexagonal shape. That's definitely one feature. You'll see that there's repeated patterns on this. That's another common feature that we'll come back to. Jonah, you seem to be...
Top left.
Why top left?
This still has that circular shape, but then it looks like it has something around it like that.
Yeah, and in fact, between the two, the one that Jonah's given is actually the better of the two answers. So he's noticed that there's kind of this circular structure here, but then there's something else surrounding this. What is this?
That's the envelope.
That's the envelope. What's the envelope made out of?
Lipid bilayer.
Phospholipid bilayer from the host, exactly. So this is going to be produced by the host. The viruses that have envelope are going to gain these upon exit from the host cell. And this is something we'll come back to later to talk about how that envelope gets formed. One of the tough things with viruses is we can only view them under electron microscopy. Or there are newer techniques like cryo-electron microscopy that help to give a bit more better resolution images. But we kind of have to use our imagination here. Okay. There are other examples. This one here would be representative of what kind of morphology?
Helical.
That's a helical. How about this one? I may have to turn off the light, maybe, or I'll kind of outline it real quick. So you've got structure here, and then I call this one, it has a dog bone on the inside. That would be a complex with an envelope. That's smallpox. Okay. This one up here is actually it looks like a bullet. It has a bullet morphology to it. This one you might consider to be complex as well. The only ones that I'm expecting you'd be able to draw are helical and icosahedral. And if it doesn't look like one of those, then we're just going to go ahead and consider it complex. There are others that are pleomorphic, ones that don't really necessarily have so much of a defined shape, but might take on 2 or 3 different types of shape. Influenza is an example of that kind of virus, where sometimes they might look spherical, sometimes they might look a little bit more oblong. Questions on the shaping here of these different ones, or just the information we've covered so far?
What would you say the top, middle?
Yeah, that one's a little bit more challenging to tell, isn't it? I think, if I'm not mistaken, it's helical. Sometimes folks would refer to a helical as being filamentous. So it looks like it's got that morphology, not as well defined as in this picture on the bottom left-hand side. If I'm not mistaken, this one might also have an envelope, Jonah, which might make it a little bit hard to decipher the actual morphology. All right, so let's take a look at cartoon images of capsids. Capsids themselves, icosahedral, helical, are the major two. They're made up of repeating patterns. So I like to think of, if any of you have seen M.C. Escher, I don't know if that's a thing anymore. It was back in my day, but so M.C. Escher, or if you ever had a geometry class in high school where you had to create tessellations and show repeating patterns, that's what a capsid is. It's a repeating pattern of either one or multiple different proteins that come together, self-assemble to form this three-dimensional structure. In this image here, what we have is a icosahedral capsid. These capsids have 20 faces, kind of look like a soccer ball, kind of look like a sphere. As they become larger, they look more spherical. And in this particular one, we have a capsomere, which is the repeating pattern of, in this case, 3 protomers, or capsid proteins. This is an example of the structure of poliovirus. Okay. Poliovirus has 3 proteins. Viral protein 1, 2, and 3. Again, those are the protomers. The 3 of them self-assemble, come together to form a capsomer. And then in this particular one, there are 60 capsomers repeated to create this structure. I'm not expecting you to memorize those numbers. Okay? Typically, we find that there is, at least with these smaller ones, smaller viruses, there's about 60 capsomers. HPV has 72. The number is not that important. Probably what's more important to know is as that number increases, that means the size of the virus is increased, or the variant is increasing as well. As that size increases, that means that the virus can hold more genomic material. Okay, so larger viruses, ones that we consider to be a couple hundred nanometers in diameter, are going to be ones that also have a larger genome, typically. Okay, I've kind of pointed out some of this already, but I just want to kind of reiterate a couple of things here. Here are 3 different types of images that we'll look at throughout the semester. Cartoon image of an icosahedral, electron micrograph, kind of looks ghost-y, and then 3D computer rendition based on electron micrograph, X-ray crystallography, etc. This probably gives us the most pure or detailed look at what these viral structures look like, but of course is computer rendered, so I just want to point that out real quick. This one is, again, 3 examples of poliovirus. One of the things I want to point out here is in this particular image, this is kind of like a topographical map in some ways. The yellow means that it's furthest out from the center. The darker blue means that this is kind of more like a valley, I suppose, in between 2 ridges. There's some importance to that. Actually, that ridge is where polio will attach to a host receptor. And the protection of that area allows, or kind of prevents the immune system from producing neutralizing antibodies that would prevent the attachment of the virus to the host. So there's some hindrance there in this structural feature that prevents immune detection. I mentioned that size here in terms of viral particles. As the viral particle increases, typically so does the genome. We can define, at least in icosahedral viruses, we can define size by what's known as a T number. So if you go on to Viral Zone and you're a true virologist, somebody who's looking at, well, what's the size of the genome, you might find what's known as the T number. There's a very mathematical way to identify the T number. I think that there's also a much simpler way, though, and the simple way is just saying how many proteins actually contribute, how many proteins contribute to a capsomer. So in this case, again, we're looking at polio VP1, 2, and 3. It's a heterotrimer, meaning that there are 3 proteins, all of them are different from one another, but total there are 3 proteins that are contributing to the structure of this capsomer. And so in this case, the T number would equal 3. We'll come back to this in a couple of minutes. I've got an example for us to try and work through. Helical capsids, on the other hand, some viruses with a helical structure will be more straight. Others, like Ebola, can actually be a bit more bendy, a bit more filamentous, but all have a basic structure. We find these associated with single-stranded RNA viruses, either plus or minus single-stranded RNA. A couple of reasons for that. One, single-stranded nucleic acid is more prone to damage and destruction than is double-stranded nucleic acid. It still adopts a helical structure, and actually that fits really well with these particular capsids. In this example here, we have 2 proteins making up a capsomer, and these capsomers are repeated throughout. Okay? We don't associate T numbers typically with helical capsids. And in this example here, you can see how that RNA is winded inside or internally of this helical capsid here, and that's the protection mechanism. Another way that we can look at this is we can look at using different computer programs, we can take a look at what is the structure of the virus or the virion with the capsid and without the capsid present. There we go. So on the right-hand side is an image of a helical capsid. The blue represents the sugar phosphate backbone of nucleic acid, in this case of a plus single-stranded RNA molecule. And you can see that the 2 of them very well overlap each other. They have the same sort of conformation. One of the important things to bring up here, and something that we'll get into in the next, a little bit later today, but even so on Friday, is we have to start thinking about this molecularly. In other words, we have to start thinking about things like charges. What are going to hold molecules together? What are going to help molecules to interact with one another? So the sugar phosphate backbone. What are some properties, I'm sorry we're going to genetics, but what are some properties of the sugar phosphate backbone? We have to understand. We'll call this molecular genetics. It's different.
Negatively charged?
Phosphates are negatively charged. That's the big one. How about sugars? They're not necessarily charged, but they can form hydrogen bonds, right? In other words, they have partial positives and partial negative charges. The importance of this, then, is that the internal face, the parts of the protomers, or those capsid proteins that are interacting with the sugar phosphate backbone, they have to have a complementary charge to be able to create a self-assembled, stable structure to protect that RNA. Okay? So if phosphates are negatively charged, then what kind of amino acids, and you don't need to give specific examples, but what kind of amino acids would we expect to be lining the inside face of these capsids? Positively charged ones with maybe partial positive charges, something to help allow for those proteins to interact with the nucleic acid. And we'll find this again and again and again. think of it as if there are two magnets drawing each other together. These partial positive and partial negative charges, or in some cases hydrophobic interactions, are what's going to drive the formation of these viral particles. Questions on that concept? All right. We've already covered this. Michaela pointed out that these envelopes are phospholipid bilayers. They come from the host. We'll talk later on about how these actually become part of that viral particle. In the case of these envelopes, we will find the receptors embedded in them. So in cases of enveloped viruses, those receptors or receptor binding proteins or spikes are typically glycoproteins, or we can call them lipoproteins. They're bound within the membrane. And then there are the complex, those that don't necessarily fit any other conformations. So we already pointed out this one, the dog bone one, that's smallpox. And then this one on the left, which I like to call the M&M with the candy corn center, okay, that's HIV. But it sounds so much more pretty as the M&M with the candy corn center. All right, let's take a few minutes to review and to kind of break so that I don't have to continue talking right now. What I want you to do is, here is a viral capsid for a herpes virus. Answer the 4 questions. How many protomers make up one capsimer? Based on, I guess the second question is a little bit more challenging, but so don't worry so much about answering the second one there. But we'll just say that the answer is 60. What's the T number? And then how many protomers are in a single capsid? So take some time, work with, feel free to work by yourself or work with somebody else that's sitting near you and work through these questions, and then we'll report out.
Yeah, yeah.
All right, let's go through and we'll kind of walk through this one. So I think I heard most people say 16 is the number in here, and I think that's correct. Herpes virus, the T number is, well, I just gave away the answer. What's the T number? 16, equal to the number of protomers that make up a single capsimer. Okay. One other thing that I want to point out, sometimes you're going to see this capsimer as capsomere. Even the book that we're using has actually gone back and forth in the terminology it's used over the past couple of editions. I do want to point out where these are within this viral structure. So you can see based on the drawing, we've got actually 2 different proteins that are making up, 2 distinct proteins that are making up this capsomere. So we've got one kind of in this teal color, and then 15 more in this greenish color. And if you kind of take a look at the image on the right, those 5 teal ones will come together as a point. Okay, those, if I'm not mistaken, those might make up the. No, I probably am mistaken. Never mind. Don't listen to me. We'll just stop there. Okay. One of the things that we'll see with viruses commonly is this feature that 5 capsomers are going to come together. Yeah, I screwed up now. There we go. 5 capsomers are going to come together to create some sort of pentagonal point or structure. Okay, that seems to be a very common feature that we find when looking at viruses. how many, we said that there, in this case, that there are 60 capsimers, and then total number of protomers over the whole viral structure. 960, just 16 times 60, right? So 960 is our answer there. Okay. Questions on how to do this? Any points of clarification? Grace, go ahead.
So telomer is the same thing as how many protomers there are?
Yeah, and or how many protomers make up one capsimer? Okay. And again, as that number increases, typically that means a larger viral size. Also means typically a larger genome. Let me give an example to kind of point that out. HPV. So HPV has a T number of 7. Herpes simplex virus 1 has a T number of 16. Based on what we've said so far, HSV1 would have the larger genome. One of the things to point out is the size kind of increases exponentially. Even though the difference between 7 and 16 is only about twofold, it actually allows for it's more of an exponential growth in size of genomes and size of the virus as well as that T number increases. Both of these have a double-stranded DNA genome. HPVs, 8 kilobase pairs, roughly, 8 to 10. HSV, 160 kb. So a much larger genome, 20 times larger than what we find with HPV. Michaela. Oh gosh, you know what? I don't remember. I will see if I can figure that out, but I don't have a good answer to that. No, that's a great question. All right. A couple other terms I want to bring up real quick, structural and non-structural. Structural proteins are going to be those that contribute to the formation of the capsid. Non-structural are proteins that will be made once the virus enters into the host. Now, there are some proteins, maybe the easiest way to break this apart is if the protein is being brought with the virus, or with if the protein is either a part of the capsid or is being brought into the cell as the capsid is moving into the cell, then we would consider it to be a structural protein. Anything else that's made after the fact is considered non-structural. That's how we'll distinguish it right now. These structural proteins can be involved in these different parts: entry, attachment. They may help to modify the host early on in the infection, allowing for selective biosynthesis or selective replication and transcription of viral genes, and then may be involved in protecting the nucleic acids as well. The other thing to bring up is that viral proteins are quite multifunctional. We're going to find proteins that function not only as a protease, an enzyme that can break down other proteins, but also function as a capsid protein. Okay? So we'll find this multifunctional feature commonly, and this allows for viruses. Again, one of the features is these genomes are incredibly small in size, or are limited to only the most essential genes for viral replication. And so having multifunctional proteins cuts down on how much genomic material needs to be packaged. Okay, I think we've talked about these other parts, so I'm going to kind of skip this slide for now. I do want to talk about one more thing today, and that's self-assembly. Probably the best way to model self-assembly is think about if you make your own salad dressing, or if you have vinaigrette at home, all right? vinaigrette is vinegar and some sort of oil, usually olive oil or something like that. You take it, you shake them up together, and what happens when oil and vinegar get mixed? They do separate over time, right? But what about right away? What happens to the oil in that suspension of vinegar? Does it mix well? No. What does the oil do?
Like little bubbles.
Little bubbles or little balls, right? So what we have here is something that is hydrophilic, something that is hydrophobic, and we have the hydrophobic, basically an exclusion property. Hydrophobic molecules will group together to exclude hydrophilic molecules, or in this case, vinegar and water. Okay? And this is an example of that. So this here is showing, here's a bunch of phospholipids, in this case, with a polar head, non-polar tails. You take this, you put it into an aqueous solution, and what you're going to get are what we call myceles. So hydrophobic tails interact with each other. This is a three-dimensional structure, like a sphere. And then those polar head groups can interact with the aqueous solution. This is self-assembly. And it requires, in this case, very little energy for self-assembly to occur. The reason why this happens is because in this conformation, we're in the spherical conformation where all of these hydrophobic tails are interacting with each other and excluding their interaction with an aqueous solution. That's a much more stable structure than having each of those hydrophobic molecules individual from one another. The properties here, this property of self-assembly and stability, is what drives the formation of capsids. So take, again, the example of poliovirus. We said that there are 3 proteins here, VP1, VP2, and VP3. All of those 3 proteins will, over time, self-assemble. They are more stable when bound together than they are individually. So we have to go back and think about what drives the interactions between these proteins, or between VP1, VP2, and VP3, or other capsid proteins. And we go back to Gen Chem, Gen Bio, Micro, etc. Biochemistry. Okay. So the properties that drive, in other words, protein-protein interaction, quaternary structures, are the same that drive the three-dimensional form of a protein. What kind of interactions are we talking about? We can go back to the phospholipid example that we looked at. We said interactions between the negative charge on the phospholipid backbone and positive charged amino acids. So ionic bonds can drive these three-dimensional shapes. Disulfide we're not going to find in the interactions that we're looking at. This would be limited to just the structure of an individual polypeptide. Hydrogen bonding. Partial positive and partial negative charges are going to drive these interactions. And then finally, hydrophobic interactions, excluding water or other hydrophilic molecules, van der Waals forces, will drive the formation of these capsids. This is a really important principle because what this means is that the formation of a capsid requires no energy other than heat to allow for these individual capsids or individual capsid proteins, the protomers, to basically bounce into each other, to diffuse into each other, and when they come into contact with each other, they will stay together more often than we'll find them independently. Okay. So, if this works, all right.
Utilizing solid printed models.
We don't have to listen to this, but I think that this, oh, you guys can't even see this, son of a gun. There we go. Okay. So here's a model that somebody created to modify this. They basically 3D printed individual capsomers. Okay, imagine the jar as the cell. Huzzah! Okay? So in this case here, obviously in this model, it's magnets driving the interaction, but you can view that as being partial positive and partial negative charges, or full positive and full negative charges that are driving the formation of these capsids. The jar represents the cell, the shaking represents, in this case, heat. Okay? And all of those three things then, the close space, so the concentration of those capsid proteins, or in this case the capsomers, the interactions between them, and the heat or the energy being put into the system all drive capsid formation. Completely beautiful randomness. Let me give you one more example of where this principle is actually really important right now. Let me see if I can find the picture I had yesterday. That one will work. Okay. If you've had the HPV vaccine, okay, HPV vaccine is what we call a viral-like particle. It is one single protein, the HPV L1 protein, which we'll talk about later, and it self-assembles. We can grow this protein in yeast, purify the protein, give it some heat, and what we will find is self-assembly of viral-like particles, essentially the capsid shell of the capsid shell of HPV. The vaccine itself has no viral genome. It also lacks the other minor capsid protein, which is known as L2. But it seems that L2 isn't needed for this self-assembly to occur. So when getting the HPV vaccine, this is what's actually going into you. The benefit of this, the host recognizes or becomes trained to recognize the three-dimensional conformation of the viral capsid, therefore allowing it to, in the long run, prevent attachment and entry of the virus into host cells. That's the design. This is kind of a unique situation, it seems like. We can form viral-like particles with other viruses as well, but HPV, I think we've just had the most success with so far. All right, two more things. One, Blackberry virus Y. I found this one. I thought it was cool. Bramby virus is the technical name. Describe the structure as best as you can. Take a minute. Tell somebody else. Share your knowledge. I hear people saying helical. Good. What's the length? Oh, see the scale bar on there.
700 nanometers.
Not far off. Yeah, that's about right. So roughly 700, 800, I think, is the typical size. Diameter? Skinny. 10 to 15. This one looks kind of 15 to 20-ish based on the scale bar there. Still filterable, though. Even though it's long, it's skinny. And that dimension there allows it to be filterable. Okay? One of the things, I guess there were three things. One more thing I wanted to bring up related to this. So Shania emailed me yesterday about one of the review questions that I put up there on for Monday's lecture, and it said, why does Mimi virus not fit the general characteristics of a virus? Now, we have not studied Mimi virus, we will not study Mimi virus. The one piece of information in the problem was, it's 750 nanometers in diameter. What does that tell us? Why does it not fit with what we know about viruses? Not filterable, exactly. So I bring this one up because for those of you who haven't had me before, those are the kinds of questions I'll ask on exams. They're going to be ones probably related to something that seem like we've never talked about it, but the information is there for you to solve the problem based on what we have talked about. Okay? You don't need to know a dang thing about Mimi virus to be able to answer that question. The only information you need to know is size and how that relates to general characteristics of viruses. Okay? Annie?
What's like the cutoff? Like, what's the filter size?
We can create a filter size. So typically, the ones that we use in our lab, I say like 0.45 micrometers, so 450 nanometers is kind of the cutoff that we use to separate bacteria from viruses. In terms of human viruses, usually the largest that we see are maybe 200 to 300 nanometers, so 0.3 micrometers in diameter, roughly. If you have questions, you can feel free to email me, but another way that I'm going to try to encourage you guys to ask questions is I put up on Blackboard a discussion board that I want to use as a way so that if you have a question, post it here, because it's probably that other people will have the same question. If you post it, it goes directly to my email, so I'll see right away what the question is. And so then we can just have a conversation through here. So what we just talked about, I put up in here. So mine and Shania's conversation over Mimi virus and this problem in particular. Okay? So feel free to use this. The last thing I want to point out, Friday's assignment is a video. And I've gone through the process of doing this myself, okay? So Brendan's already